Okay. Okay. But
Hello, Quentin.
Hello.
Hello. How are you?
Good, thank you.
What time is it there?
It's 7 p.m.
7
So
p.m.
early evening, a perfect time.
Thank you so much.
So I'm just so can I don't know whether you can see my screen.
Yes.
All right, I'll just change to presentation mode. So is Marlena there?
Sorry.
Marlena? Oh, I
Yes.
just wanted to say thank you for the Excelso coffee, which is going to be delicious. I haven't tried it yet, but I'm really looking forward to it.
My pleasure, Quentin. Jenny is not around now.
Apparently, Seni is um, in in the country. She's away from Vancouver in the mountains. Um, but she said she would try to attend if
Okay.
she had a signal. But she
Yes.
might not have cell phone signal in the
Yes.
mountains.
Selamat pagi Bapak Ibu semua. Selamat pagi Mbak Marlina. Apakah suara saya terdengar?
Ya, terdengar Gloria.
Oh, oke, siap.
Oke, sip. Terima kasih.
Ya, kita tunggu beberapa menit lagi ya Bapak Ibu, mohon kesediaannya. Menunggu mungkin Pak Dul untuk memberikan sambutan. Sepertinya belum join.
Oke, nggak apa-apa, kita tunggu aja.
Ya, oke, siap. Terima kasih.
Hello, Quentin. This is Alex.
Hello.
How are you?
Hello, Alex. Very, very well, thank you.
Yeah, well, sorry about the time arrangement, but this is the best we can.
Yeah. No, it's a good time.
match.
Good time for me.
Yeah, so 9 a.m. here in Jakarta and 7 p.m. in Vancouver.
That's right. Yeah.
And I've been reading your CV like 8 pages. That's something. I don't know how to make a proper introduction.
I've been around a long time. That's all that means.
And yeah, I also noticed that you were in Oxford,
Yes.
in the Department of Plant Sciences, and
That's right. I was a departmental lecturer in Oxford in the early 90s.
yeah, in the early 90s. Yeah, because
And
I was
then-
there in 2016.
Right.
I was supervised by Robert Scotland.
Yes. Well, I know Robert, of course.
Yeah. So, well, we share the same route, a common ground in England.
Yes. Yeah. So after Oxford, I left Oxford to go to the Royal Botanic Garden, Edinburgh. Of course, is a very famous.
Oh yes, indeed. I think
botanical
that's the
place.
second oldest, or is it the oldest one?
It is very, very. I'm not sure it's the oldest, but it is very old. So I had a very happy 7 years in Edinburgh before accepting a chair at UBC in Vancouver. But in Edinburgh, I worked on Gesneriaceae and Zingiberaceae, which is how I met Marlina.
Yeah, Zingiberaceae and Gesneriaceae. Well, those two are tough.
Yes.
Yeah, and today we are going to talk about flowers. That's interesting.
Exactly.
Yeah.
Excuse me.
Yeah. No, yeah. Go ahead.
Sorry for interrupting. Yes. Shall we start now? Because Pak Dul is already here. Ya. Ya, oke. Saya buka ya Bapak Ibu. Selamat pagi, selamat datang di acara Session of Sharing yang ke-69. Hari ini kita akan mengikuti kegiatan SOS yang akan dipimpin oleh Kelris Monokotil. Untuk sebelum kita memulai dan saya serahkan ke moderator, saya minta kepada Pak Dul selaku PLH PRBE hari ini untuk membuka acara atau memberikan sambutannya. Kepada Bapak Dul dipersilahkan.
Baik, terima kasih Mbak Gloria. Okay. So, selamat pagi, good morning everyone. Staff of Research Center for Biosystematic and Evolution BRIN and other research center under BRIN maybe here, or anyone else audience outside BRIN. And also, good evening to Professor Quentin Kram. I think it's late night there in Vancouver, isn't it? I'm sorry, we have to take your times. You have probably need to rest in this tonight, but we ask you to present some here in this. Okay.
It's a great pleasure to be here and a great honor to have been asked.
Okay, so very warm welcome here to all of you, and thank you for joining this 69th season of sharing for the Research Center of Biosystematic and Evolution, BRIN, or National Research and Innovation Agency. This is our regular scientific meeting to watch and listen any updating for the scientific knowledge of biosystematic, evolution, biogeography, and other related topics. So everyone, today we will watch and listen presentation from our college. So this is actually not unfamiliar person for me. So Professor Quentin Krong from the BD Biodiversity Museum, University of British Columbia, Vancouver in Canada. So I heard many times from Professor Krong or Quentin, that I realized that you were previously in the UK, isn't it? So
That's right.
and we know that some of us know that you are now a supervisor for our staff, Seni Kurnia Sanjaya, and also collaborator for Dr. Marlina Diani here for some project or research project for the Shinji Bresi. So
That's right.
yeah. Thank you for spending your time here. And Professor Krong will share his knowledge here about plant evolution in general with us, with the title of the plant sexual system from hermaproditism to dioecy. So I think this topic is great and very relative to our main research here in Research Central, Biosystematic Evolution. So especially for our staff, we mostly work for plants evolution. So I also read from the abstract. Professor Krong explained about what he will present for today, that are how the plants evolve their reproductive system from bisexual into unisexual from the genomic and evo-devo perspective. So this is really interesting topic. So I hope this morning or today season of sharing running smoothly, and there will be fruitful discussion after it. And also, we hope that this valuable sharing can be useful for us for our next research, especially when dealing with evolution, not only for plants but also for animals and microbes and so on. So I think not for long again my speech, so I give back to the moderator, and hopefully we can watch and listen this interesting representation and decision. Thank you very much. So.
So are we ready for me to begin?
Oke, hang on. Gloria, langsung aja atau gimana?
Ya, terima kasih Pak Du untuk sambutannya. Ya, saya serahkan ke Pak Alex selaku moderator. Terima kasih.
Oke, terima kasih teman-teman, kolega, Bapak, Ibu sekalian yang ada di dalam ruangan dan di luar ruangan. Terima kasih telah bergabung dengan SOS seri yang ke-69 ini. Sekali lagi kita di sini Pusat Riset Biosistematik dan Evolusi, kita fokus kepada sistematik dan juga evolusi, dan kita berusaha menampilkan jajaran pembicara dengan latar belakang yang bervariasi, namun tetap di topik yaitu sistematik dan evolusi. Dan karena kali ini kita kedatangan Profesor Quentin Krong dari Kanada, saya akan menggunakan bahasa Inggris dalam memoderasi ini. Okay, again, thank you again for joining us, Quentin. I just introduce you. I will introduce you to the audience online and offline. And previously, the acting director Abdul Rahman Kartonegoro has welcomed you, and we are really keen to have a continuing cooperation with you, especially with Danny as the meeting points. And I'll try to make the introduction short because having looked at your presentation, that's a lot of muscle for for the presentation. And ladies and gentlemen, right now we have our presenter, Professor Quentin Krong. He's a botanist, plant taxonomist, and also dealing with genomics and biodiversity informatics. So that's a whole range of disciplines in one person. And speaking about his career at the moment, he is in University of British Columbia in Canada, and he's a professor there. And also he is the director for BT Biodiversity Museum. And it's not really easy to like compress 8 pages of your CV in this proper introduction, but I'll try my best. So as I mentioned before, that Quentin is a professor. At the moment, he's supervising students from Oxford, Edinburgh, as well as University of British Columbia, or UBC. And his achievement, his academic track is amazingly good, because I think, the fact that you were in 3 cities with amazing achievement in academics, like you started your undergrad in Cambridge, and you get your bachelor and master's there, and you finish up with PhD in Cambridge, and I think you are dealing a lot with plant life of Saint Helena. Would be interesting to to see them. And after Cambridge, you went to Oxford, to the Department of Plant Sciences, the very same department I did my DPhil, and it's really an honor for me to host another Oxonian. So again, welcome. And you were the director of, the curator of Oxford Herbarium, Oxford University Herbarium, and while in Oxford, Quentin worked a lot with great names like Richard Dawkins and David Mabely to nourish the next generations of biologists. And afterwards, you went to Edinburgh. Well, we know Edinburgh is very famous with people in biology, especially like Charles Darwin. And in Edinburgh, you also involved with the Royal Botanical Garden of Edinburgh. And afterwards, you also currently dealing with editorships with systematic botany, as well as Plant People and Plant Journal. Those 2 journals are highly respected journal in botany, and that's very prestigious position. And for your publication, for Quentin's publication, he has published a whole lot of publications, especially in Nature, Science, as well as PNAS. And of course, you get a lot of funding from different and various professional associations and communities. So that must be really something to have you here. And hang on, after, I think I don't want to take too much of your time, because you will present something that is really interesting for us. Our basic instinct, the thing we cannot live without, the sex, or well, bear with me. In this case is the plant sexual system. And before I give it the platform to you, I will also acknowledge some audience, because we have audience from inside and outside of Indonesia. We have Kunman Wong, as well as other people outside of BRIN. And well, thank you for joining us in this session. So Quentin, it's all yours. The platform is yours for, I think we're gonna have like a presentation for like 40 to 50 minutes, and we can continue with Q&A. Thank you.
Right. Thank you, Alex, for that very generous introduction. Please let me know if you can't hear me, but I'll assume that you can hear me well if I don't hear from you. So I'm going to talk today about something that has interested me very much for a long time. And I'm not the first person to be interested in this. Charles Darwin was extremely interested in it, as we'll see later. But first of all, I just want to give you a refresher on some of the most common systems. There are many, many plant sexual systems, but these are the commonest. And just for those who are not botanists who are in the audience, we have most flowers. Most species of plants have hermaphrodite flowers, by which we mean, as botanists, that they have the male and female organs very closely together, or fairly closely together, in a single flower. But this can change, and on the right-hand side, we have dioecy, where the different individuals have different sexes of flower. Now, I'm showing a picture there of a hibiscus flower, and it's really just to mention that we can divide very simply a flower into male function, which I've labeled M, female function, which I've labeled F, and attractiveness, which I've labeled A. And this is typical of insect-pollinated flowers. And if we ask the question, why are bisexual, or as we call them in botany, hermaphrodite flowers so great? Because the vast majority of flowers are hermaphrodite, like the hibiscus at the top. And we can answer this question using a resource allocation argument. And it's really the only argument that's ever been put forward. We assume it's right, but we have really no experimental proof that this is the reason. But the resource allocation argument goes as follows. So hermaphrodite flowers, each flower has male function, female function, and attraction function. And all these have a cost. So you have a cost in petals, or a cost in carpels, or a cost in stamens. But they're bundled together. Each flower has M, F, A as costs. If we separate the sexes, as we see in that cucumber flower at the bottom, the yellow flower is a cucumber, then some flowers just have M and some flowers just have F, but they both have to have A. So if we just add up what the cost is, the cost to the plant of hermaphrodite flowers is M plus F plus A. But the cost to the plant of the same amount of function spread over two flowers is M plus A and F plus A. So it's M plus F plus 2A. So the cost, there's twice as much cost in producing organs of attraction, the petals, and indeed of protection, the sepals. So I think you can see that how this argument works. It's more efficient to bundle everything together as a single flower. And that's how the argument runs. And resource allocation models, I think, are very powerful and interesting. They haven't been developed to any great extent in a lot of areas, and I think they could be much more developed. But we can see that there are many ways a plant can organize its sexual function. It can produce, as in that tropical rainforest tree, many, many thousands of very small flowers, or in the case of the giant water lily, a few very large flowers. And this is a resource allocation thing. It's the evolution of resource allocation. And I think it would be a very interesting topic for someone to explore in more detail using mathematical models. Now, this leads us on to a problem. If hermaphrodite flowers are so beneficial, so efficient, little cost, how do unisexual flowers and dioecy evolve? Now, in 1978, Charlesworth and Charlesworth tackled this question. And you can see, I don't know whether you can read this down here, but they have a quote from, they have a quote from Charles Darwin to lead their paper. There is much difficulty in understanding why hermaphrodite plants should ever have been rendered dioecious. This is a quote from Charles Darwin in 1877. And they set out to solve this. And they produced a model which is very famous and until recently has never been challenged. And basically it involves two important equations. There are many equations involved in this argument, but there are two very important equations. These are equations which govern the invasion of a male sterility mutation and a female sterility mutation. So they argued that it was important to have a mutation that removed stamens from flowers and a mutation that removed pistils from flowers to get dioecy. So they made some equations which related what can be called seed compensation, K, which is the extra benefit to the seed production of less selfing, more outcrossing, which you'd expect from dioecy. And K may be more ovules, or it may be higher quality ovules, less inbred ovules, but it's seed compensation. And here they relate it, the equation relates it to inbreeding depression and selfing rate. So the argument is all about preventing inbreeding selfing. And then for the other, then once you have the male sterile mutation invading, you have to have a female suppressing mutation to invade, turning the remaining hermaphrodites more male-like. And again, they have an equation which relates the loss of ovules, seeds, to inbreeding depression and selfing rate. I'm not going to go into these equations in any detail. That's not the remit of this talk, but I wanted you to understand where the current theory comes from. And it's been with us since 1978. But I've always thought there are problems with the Charlesworth theory. So the Charlesworth theory really is about dioecy evolving via gynodioecy. And there's a problem with that. The first problem is that most dioecy evolves via monoecy and not gynodioecy. And conceptually, one can imagine scenarios where the evolution of dioecy from monoecy can involve only one gene and doesn't need two genes. It's going to need two mutations, but at the sex determining region, it can be one gene that acts as a switch that segregates. It's either the switch is present or the switch is absent. And that's intuitively quite clear. It's always been clear to me, but it's sometimes such has been the dominance of the two-gene model that has been very difficult for people to accept. So we've really needed an alternative theory to model sex determination involving just a single switch gene. And we were at that point, we knew we would need some evo-devo and genomics to find what the sex determination really is in different systems. Is it two linked loci, as the Charlesworth theory would state, or is it just one locus, one gene? Theory is not enough. We need to go into the lab. So many years ago now, I started a project on the Salicaceae. And there are many models for sex determination. A model for monoecy, one of them might be Zea mays, a grass, because many grasses are monoecious. And the grass system is extremely interesting, and how that evolves, and much is known because of Zea mays, the maize plant. But we wanted to look at dioecy, and we knew that in the Salicaceae, dioecy evolves from monoecy. So this seemed to be a good example. Now, we used a model tree that's common in Canada. That's poplar or populus. It's not present, I don't think, in Indonesia, but you have its sister genus Salix, Salix tetrasperma. The botanist in the audience will probably know Salix tetrasperma, one member of the Salicaceae. And actually, many Salicaceae are dioecious in Indonesia, such as the genus Casiaria, which is a common tropical forest genus. But we used Populus because there was a good genome, or what we thought was a good genome. But it took us a long time to get the answer. The first thing we did was a GWAS. That stands for Genome-Wide Association Study. So we took a lot of males and a lot of females and asked the question, what genomic regions are associated with being male or being female? And which is a process called GWAS. We had that generated some candidate genes. Then we needed to know whether those candidate genes were expressed differentially. So then we used transcriptomics. Then we suspected that the critical gene was controlled by methylation. It was epigenomic. And so we did a genome-wide methylomic study. We also found that the genome wasn't well assembled and that this was causing a lot of problems. So my colleague in Germany, Niels Müller, undertook to reassemble the chromosome responsible for sex determination using long read sequencing. And we also needed to do sRNA, small RNA sequencing, because it turned out that the mechanism involved small interfering RNA. So all these things led up to a functional model for how sex was determined in poplar. But still, we needed experimental proof. And my colleague in Germany, Niels Müller, and the group at his institute managed to knock out what we thought was the key gene, and by CRISPR-Cas9 technology, and we were able to change sex. But let me go through this in a little more detail. So first of all, we did the association study. And if you do an association study, a whole genome association study, you get diagrams a bit like this. So these peaks are where there's a gene or a series of SNPs, single nucleotide polymorphisms, that are associated with sex. And to our consternation, these were spread out all over the genome. We tried it on one species, then combined one species, then another species, then we combined the two species, and still the association with sex was in different places. And this was very puzzling because how can you have different chromosomes all being associated with being male or female? These chromosomes will segregate independently. So we knew that something was wrong, and it turned out that the genome was actually badly assembled. It was at the time, it was one of the best assembled genomes we had, plant genomes that we had, but it still had problems. And I like to warn people about genome quality because this image shows it very well. This is the genome we want, the perfect horse. But this is the genome we get because many genomes are assembled or used to be assembled using short read technology. And with short reads, you assemble contigs, and it is very difficult to put these contigs together reliably. So this is our genome. But the genome we want is this. And so this is just a warning. However, it's getting better and better. And I have started some work on the Urticaceae, and we've generated some genomes which, using long read technology, and high C technology, which we believe are very, very reliable indeed, and assembled in chromosome length chunks from telomere to telomere. So one thing we noticed, our candidate gene is this gene ARR17. And one thing we noticed was that this gene is methylated in males and highly methylated in males, but unmethylated in females. This is at the promoter region. So the promoter of the gene is methylated in males. So this gene is not expressed in males. So it's a feminizing gene, but we still faced the problem of how was sex determination working in this system. We had an idea that this was the feminizing gene, but it's present in both males and females. It's just being methylated and suppressed in males. So it's definitely a feminizing gene. But how does this actually work? And when we assembled the genome and overlaid the male and female reads, we find that the male and female reads over the length of this chromosome, chromosome 19, they track each other. So you can see the blue here, the blue line and the red line are tracking each other almost perfectly. And but here, where ARR17 is, there's more blue. So we knew there must be some kind of duplication. We were getting extra copies from somewhere. And it turned out that the duplications were somewhere else. So ARR17 was down here, but the duplicates were up here. But the duplicates were being assembled on top of the gene that they're duplicated from. And these duplicates are facing each other. They're inverted duplicates. And inverted duplicates, when we saw that, we knew what was going on, because inverted duplicates create double-stranded RNA. And double-stranded RNA is immediately cut up by a protein called dioecy into small RNAs. And these small RNAs can interfere with the expression of genes. And what the model we came up to is that these inverted repeats with the same sequence as our feminizing gene, these were only present in the male. And they produced small RNAs which silenced the feminizer using RNA-directed DNA methylation, or RDDM, as it's called. So it's a very beautiful mechanism. So it wasn't that the feminizer was important in determining sex, but it was actually being controlled by the presence or absence of this switch. The presence of these inverted repeats in the male was switching off the feminizer. Very beautiful. And we were able to publish this with Niels Muller as lead author in 2020. And then we had in that same paper an experimental proof. So this is a female plant. And normally a female plant will have, which is XX, will have pistils like this. But if you knock out a single gene, ARR17, which is the feminizer, just one gene, you get a perfect male, an XX male. Ought to be a female, but because one gene has been deleted, it turns into a male. And not only that, but this male has perfectly good pollen, and can be used in breeding experiments. It's just a normal male. So this was experimental proof that it is possible to determine sex with a single gene. You didn't need two genes in linkage. So this was directly against the Charlesworth theory. So now we have experimental proof of single locus, single gene sex determination. Will theoretical biologists now update theory? And the answer was yes. After our paper was published in 2024, Michael Scott and Simone Imler came up with what is a very important paper, which is redoing the theory to show how one-factor sex determination can evolve without linkage between two genes. So we now have new theory to back up the experimental results, which is very nice. So that's just an example of how it's possible to use genomics and laboratory biology to determine how sex is controlled in dioecious plants. But where are the dioecious plants? Where are our future research subjects? And they're actually very common. And because I'm giving this lecture in Indonesia, I'd point out that tropical rainforest biology is full of dioecious plants. Of course, figs, which are very common in tropical rainforests, have a very interesting sexual system with functionally dioecious types, which are still actually monoecious at one level. And I can also give example of mangoes, mangifera, and I've just been to Singapore, and in Singapore there are these very common species which are also either dioecious or monoecious. And also, when I gave a lecture recently at the National University of Singapore, they were very interested in this plant, Cocculus orbiculatus, which is very, very common and widespread plant, but it's very rare in Singapore. And so they're very interested in conservation of this plant, even though it's common elsewhere. But in Singapore, only male individuals are left. So it is a big problem for conservation because it's dioecious and the population is just males. So I say this because there are practical reasons for studying the sexual system of plants. There are conservation reasons, there are agricultural reasons, and there are reasons to do with one can learn so much about fundamental biology in studying this topic. So I'm going to now separate out further work into some tasks. So one thing that would be very helpful is an accurate description of the sexual system. Now, I don't want to blame taxonomists because I'm a taxonomist myself, but I will say that taxonomists could do a better job of describing the sexual system of plants. Very often in descriptions, the differences between male and female plants, or even the form that dioecy or monoecy takes, or in more complex sexual systems, is often glossed over in taxonomic descriptions. And it'd be much better if there was some detailed descriptive work. And recently I was invited to give a lecture in Brazil by some Brazilian botanists who were interested in leguminosae. And many mimosoid legumes in particular, but also some cisalpinioid legumes as well, are dioecious or monoecious. And I was asked, this question: how do you describe the sexual system of plants? So I said, well, why not write a paper? And so we wrote a paper describing how to describe plant sexual systems based on legumes, but it could apply to anything. And I proposed what I call an inflorescence formula, which is just a simple way of setting down what is happening. So this plant, some of you may know, I think it's grown as a garden plant in parts of Indonesia. It's native to Africa. Dichrostachys cinerea. It has hermaphrodite and neuter flowers. So it's a sexual difference, but a rather odd one, because some of the flowers are neuter, and then you get these intermediate flowers. So there are three floral states: hermaphrodite, intermediate, and neuter. This is very interesting because there are costs associated with having neuter flowers, but no reproductive gain. These neuter flowers are not going to be producing any seed or pollen. But as you can see, the neuter flowers are bright pink, and so obviously have attractive function. So I suggest, well, why not actually say what's happening? Don't just say at the base there are some flowers which are neuter and then intermediates, and try and describe it in words. Why not just simply jot it down? N for neuter flowers, an average of 34. This is an average of just 4 inflorescences, but you could do quantitative statistics here. Intermediates 11, hermaphrodites 117. That's in the inflorescence. And then this sign here for the apex. So this is going from base to the apex. And this is a very simple example, but we made the formula such that it could describe any sexual system, even the really complex polygamous systems that have complex mixtures of hermaphrodite, male unisexual and female unisexual flowers. And I hope it may help people to describe the way that flowers of different sexes are distributed over individuals and over inflorescences. The second task, which is important when you have monoecious species, is to calculate the variation in what's called quantitative gender. If you have dioecious species that are strict, so you have strict females and strict males, there's no problem, because the males are males and the females are females. But what happens if you have a male that produces a few female flowers, or a female that produces a few male flowers? Or if you have a monoecious species that starts out producing just male flowers and then produces a few of both and then ends up just producing female flowers. So quantitative gender is changing developmentally over time. So fortunately, the theory of this and the formula was worked out by Lloyd, a famous New Zealand biologist, in 1980. He wrote this paper, a classic paper, Sexual Strategies in Plants, number 3, a quantitative method for describing the gender of plants. And sorry, we're going. So G is functional femaleness. Functional maleness is just 1 minus G. So this is an index that goes from 1, which is fully female, to 0, which is fully male, or a hermaphrodite plant would be 0.5, for instance. So, I mean, I'm not going to describe this in detail, but basically we need some indication of female function, female investment, male investment, which is A, and then some sort of equivalence factor, because if the population has fewer male flowers generally, then male flowers are more valuable. So they have to be given an extra boost, and you do this using this equivalence factor. So using this formula, you can track the maleness and femaleness of plants between individuals, if it varies as a population level, or over developmental time, as monoecious plants vary in their gender developmentally. So there's a third task, which is, well, what's transporting the pollen? Because if we take the view that dioecy or monoecy are fundamentally have something to do with the avoidance of selfing, which isn't all that clear in the case of monoecy, but it's I think indisputable in the case of dioecy, then we need to know, well, how is this actually working? How does the system work ecologically in the ecosystem? And I'm very happy that my student, Seni Sanjaya, has spent some time recently in Sulawesi studying a monoecious ginger, Zingiberaceae. And it is clearly vertebrate pollinated, either by bats, as we can see here. There's a beautiful example of a flower visiting bat, and or by birds. So during the day, it's visited by these honey eaters. So this is the kind of information we need in order to start understanding the systems and how they're working. How is pollen being transferred from male flowers to female flowers? What aspects of the floral biology and the sexual system have evolved to fit with these pollinators? So I'm going to finish there, but I'm deliberately leaving a lot of time for discussion, because what I'd like you to do is to think about your favorite example of a plant species with floral polymorphism. It's usually unisexual versus hermaphrodite flowers, but it could be other forms of floral polymorphism. And I'd very much like to start a discussion. And also, if you have questions about particular aspects of the work that I've presented, would like me to say more about it, then please let me know, and I will talk more about those particular slides. But I would really like to hand it over now to you to come up with your favorite plants so we can discuss them.
Okay, thank you, Quentin. That's such a lovely and amazing presentation, because you provide a different perspective, like about sex, not as we know it now, with different formula, calculation, and also consideration whether it may work in evolution or not. Because when we look at monoecy and dioecy, it seems that they have to invest a lot of energy just to attract pollinators. And okay, without further ado, I'd like to open this Q&A session to the audience. We have like 82 audience online, and we have one question in chat box. So would you like me to read it out loud for you, or?
I can see it actually,
Okay.
so I can take it.
Yes.
Question to Professor Kronk: what is the implications of plant evolution from hermaphrodite to dioecy on developing effective breeding programs, like for many species, Mangifera, especially in Borneo, cucurbits, etc.? Well, that's a big question, but a surprising number of our crops, particularly woody crops, are in fact dioecious. So in some cases, I think I'd pick out maybe 5 answers to this question. One answer is that some breeders have focused on breeding monoecious or hermaphrodite flowers from dioecy. So the grapevine that makes wine and produces grapes is commonly grown in Europe and many places. Ancestrally, that is a monoecious or dioecious species, but it's been bred. People have selected hermaphrodite flowers to get more reliable grape crops. So it's been very important for people have bred for hermaphroditism now, and they bred for hermaphroditism even before we knew the molecular biology of the system. But we now know in grapevine what the genes are in quite a lot of detail. So it will be much more easy to breed for sexual systems that agriculturalists want. I would also say that in plants like maize, Zea maize, complete male steriles have been used in order to do F1 breeding. So when you buy maize seed, very often you will buy F1 plants. And that's done by manipulating the sexual system. So you have a mutant that doesn't produce any male flowers at all. And then so all the seed will be formed by cross-pollination. And by this way, you can raise F1 seed. There are many, so it's really, I think, quite useful to know the molecular biology because then you can manipulate the system. And breeders have always been manipulating the sexual system of plants, but they've done it without knowing the underlying genomics and genetics. Now we do, it should be easier because we now know what we're doing instead of doing it unconsciously. But I could go on with other examples, but I think that gives the idea.
Okay.
Sorry, we have another question in the chat.
Yes,
Shall I go through
ahead,
on the chat? So thank you for your presentation. My question is, how can you determine ARR17 as a sexual gene in those plants? And do you consider some other gene which may have the same or similar function on sexual dimorphism? Well, yes, that was the key question we had to answer. And it seemed that it was the only gene that was or the controller gene, the gene that was controlling that gene, was the only gene that was active. But to prove it, we had to do an experimental test using CRISPR to knock that gene out. And knocking out that gene changes the sex. And no other gene was knocked out. So if there was another gene, because we kept on thinking, well, maybe there's another gene, maybe there's another gene. But if there was another gene, we wouldn't see such a clean change of sex just by knocking out AR17. So that was what led us to really believe and to make other people believe that AR17 was the only gene that was responsible. So shall I go on with the chat?
Go ahead, Quentin. Yeah, the next one is from Kunman.
Right. So might you also comment on the change in sexual state of an individual plant, such as the papaya? Yeah, so of course this is very interesting. So um, sex change you can call sequential monecy. It's the plant is fundamentally monecy, but it may produce all female flowers for many years, and people think, oh, this is a female. Then it suddenly produces male flowers. So it's always had the ability to produce male flowers. It just has a long time between male and female production. And there are aroids that do this as well. The genus Aracema is well known for changing sex in this way. It's not really changing sex. It's just having both sexes, but expressing them in over a very long time scale differently. So it appears to be a sex change. And I think this is a very interesting phenomenon. It's obviously rooted in plant physiology. It needs much further study. The surface has not really been scratched. We need molecular models of what's going on to control the unisexual flowers in these species, and then to see how these genes are controlled. I think it's a completely open field. We need to know much more about it. Do you want to introduce the next question,
Yes.
Alec?
I think the next person just raised hand. Eka Iskandar, yes, please go ahead.
Hello.
Hi.
Hi, Quentin. First, I'm going to answer the last slide question about my favorite plants.
Oh, yes, please.
Okay. I'm working with bryophytes. So I'm wondering whether these genes could be also found in bryophytes in the lower plants.
Yeah. That's a very interesting question. So I didn't talk about bryophytes, but of course bryophytes have instead of an XY system, which is common in mammals and in plants, they have a UV system, what's called a UV system, because it works in the haploid phase. So in bryophytes, the sporophyte is sexless. It just produces spores. So the sex is expressed in the haploid stage, in the gametophytes. And of course, it's very common to have dioecious bryophytes and monoecious bryophytes. I think it's not my specialist subject, but I know there is some very interesting work being done at the moment by various bryologists to work out the sexual system. I did a little bit of work myself on Marcantia because Marcantia is a genus of thalloid liverwort that has, as you know, male and female gametophytes.
Yep.
So clearly has a UV system of sex determination. So as the spores, as the sporophyte forms spores, there's meiosis, and so some spores get a U chromosome, some spores get a V chromosome, and sex is determined that way. I would love to work more on the systems in bryophytes because they're absolutely fascinating. So you're very lucky to be studying bryophytes because what an interesting group they are. What if I can ask, what is your favorite bryophyte, and is it monoecious or dioecious?
Oh, I have so many favorite bryophytes, so I cannot really think. And for now, my student also just asked me, how do you make sure that this plant is monoecious or dioecious? I cannot answer that.
Yeah, I know it's all sorts of interesting details in bryophytes. So, Yeah. Thank
Okay,
you.
so maybe I
Thank you for
can
your
ask another question.
Yeah.
Yes, this is about the coculus plant that you
Oh,
found in Singapore, about that it is now a male only plants.
Mhm.
So with this technology that you have, would you then be knock out this male into the female one then for the conservation purposes?
That is exactly what they asked me in Singapore. And my
Okay.
answer was that in theory, one can do if one understood the sexual system in cocculus, one
Yes.
could potentially manipulate it using CRISPR, say, to produce some females. But
Ah.
it would be an incredibly time-consuming and expensive way of doing it, because
Yeah.
just developing transformation systems for tropical tree species would be hugely difficult. So you would probably need millions of dollars to do it that way. The easiest way is just to go to the across the strait to Malaysia and get a female. But
Yes.
see, they don't really like to do that because they say, well, it's not Singaporean. We want to conserve the Singapore flora. We don't know whether we're bringing in a different type or a different genotype, so they'd much rather find a female from Singapore or do transformation to to make Singaporean males into females.
Okay.
So, I told them just go and get one from Malaysia.
Okay, one last question. Oh, you show the picture from the Populus tremula and Populus trichocarpa.
Yeah.
So do are these sexual system species specifics or not?
Yes. So we believe that all
Oh.
poplar species use the same gene, ARR17, as a feminizer. So at that level, it's the same in all species.
Mm-mm.
But there's a but coming. But the details are slightly different in every species.
Yes.
And
Ah.
having worked it out in one species, we can then look at the system in all the other species. And the results are very interesting because some species use the system I talked about of having the feminizer gene and a suppressor, the males having a suppressor. So males are males because they suppress the feminizer. But in other cases, in other species, like the white poplar, Populus alba, they do it slightly differently in that there's only one copy of the feminizer gene. So the males just don't have the feminizer gene at all, and the females have one copy. So it's a ZW system, not an XY system. You've got it depends which sex is heterogametic.
Yeah.
So in white poplar, the female sex is heterogametic. So it's ZW, whereas in the poplars we worked on first, it was the male, it was XY. Humans are XY as well because the male has the heterogametic sex. It's the male who determines the sex of the next generation because by passing on a Y or passing on an X. And so in evolution, there'd been a flip. There's been a mutation which just meant it did it, used the same gene, but did it in a different way. I didn't really have time to talk about that, but the patterns of change between different species using the same gene but in different ways is really fascinating. And willows do it appear to use this ARR17 gene as well, but again do it in slightly different. The details are different.
Complicated.
Yes. So, but if you go to another genus or another family, then it's a different gene.
I can imagine, yes.
This has evolved independently many, many times. And every time it seems to use a different gene, which is so the take home from that is that there are many, many ways of making a male or making a female.
Yes.
I think at the level of the floral developmental genes, there's only one way. But at the level of the switch, because what we're talking about is the switch, it's like a light switch, male on, female on. At that level, the switch can be all sorts of genes. You just need a switch and to connect it to the floral developmental genes. And what we don't have at the moment is the knowledge of the pathway between the switch and the floral developmental genes, like Madsbox genes. We know that there's a connection. We just don't know the genes in between. And that's the next stage.
Okay. Thank you.
Okay, thank you. Those three questions, it takes a lot of muscles from Quentin to describe it. But that's very interesting, and thank you for the discussion. That's a very valuable one. And the next one, we have another question from ST on in chat box. And ST, I know this lady. Please feel free if you want to talk to Quentin yourself. Yeah. Otherwise, yeah, in the chat box, she's asking about specialization, gender in heterostyly. Would you like to read it?
Yeah. So heterostyly is very interesting, of course. It is a pathway to dioecy, and Darwin recognized that heterostyly could be a gateway to dioecy, because heterostyly is a dimorphism, and so it's quite possible to imagine in evolution the long style morph becoming a female and the short style morph getting the style getting shorter and shorter and shorter until it disappears and becoming female. So and we are just we're just starting to understand the genomic basis of heterostyly. The genetics has been sort of known for a long time, the idea of a supergene, but we're just starting to get the first papers actually seriously looking at the genomic basis. So gender specialization is thought to be a transitional phase from distyly to dioecy. I agree. I agree. Could climate change be a factor influencing the evolution of flower systems? For instance, Sarcotheca macrophylla in the Oxalidaceae, distyly has a different flower type, long style flower and short style flowers. The long style tends to enhance female function and have low pollen viability, and the short style flower has a great pollen viability. Yeah. So I think that's such a great system. I didn't know about that, but it really intrigued. Now you've told me, I'm really intrigued. I agree that that distyly can evolve towards dioecy, and this, from what you say, this seems to be happening in Sarcandra, and also this is a matter of where quantitative gender could come in. So long-styled morphs might still have male and female function, but they might have more female function. And one could use Lloyd's equations to say just how female they are and put a number on it. And take the short-styled morphs and say just how are they extra, are they 0.5, which you'd expect a hermaphrodite flower to be, or are they changing? So I think that's a great potential study system from what you say. So thank you for bringing that up.
Okay, for the next one, in the chat box from Mukhtar Davis. Oh, that's a lot. I think, yeah. Please take time to digest it because, yeah, like 2 paragraphs. Take your
Okay.
time.
So I'll read it out because that
Yeah.
helps me understand it. Diploid wild banana Musa acuminata, yeah, propagates vegetatively by forming clusters of clones around the original individual. However, it also reproduces sexually through pollination. The flowers are hermaphrodite. The male and female organs mature at different times, with the female organ maturing before the male. So self-pollination is very rare. So yes, so I imagine that pollination can occur either within a cluster, between closely related plants, or with distant plants from other clusters. What do you think the heterozygosity levels would generally look like in the seed batch produced by a mother plant in wild bananas? Do you have any insights from other species with similar reproductive strategies? Okay, this is so I would say with clonal plants with large patches, you need a very effective pollinator that will transfer pollen long distances to get outside the clonal patch. And really just vertebrates will do that. So I would suspect, but I don't know, but you can tell me what the pollinator is, but I would suspect that this is either Musa acuminator. Is it bird pollinated? Is it sunbird pollinated? Musa acuminator? Does anybody know?
Coba. Pisang. Nah, kita nggak ada yang tahu.
Do we know what pollinates Musa? I
Yeah,
feel I
that's
should
the
know, but
question,
I
isn't
just
it?
can't remember.
Mukhtar, oh, bat
Oh,
and
bats and birds. Yes. Well, that's exactly what I would predict because you need something that has a very long foraging range. And so a trap lining pollination strategy would work. So if you have a colony of bats, which is coming in foraging on one clonal patch and then flying several miles to the next clonal patch, then you're going to get a lot of pollen transfer between clonal patches. And so the heterozygosity could be maintained as quite high. But of course, if the pollination system fails, if deforestation reduces the bat population or the bird population, then you could get a serious inbreeding problem. So I think of this as a kind of ecological question. And it could very well be affected by factors like deforestation and the availability of vertebrate pollinators like birds and bats. But yes, that's an interesting one.
yeah, and we still have question from Kun Meng Wong. And since you have explained a lot, Quentin, let me read the question for you. So from Kun Meng Wong in chat box, he's asking whether is there any experience with sex differentiation in holoparasites like Rafflesia or Sapria, because the main plant body within or both of them have like main plant body within or intermingled with host tissue.
Yeah. So I would so much like to have the chance to study Rafflesia. Who wouldn't? This is a wonderful plant. But I don't know anything about its differentiate. Are Rafflesias? I mean, it's a huge flower, I know, but I have limited experience of. Funny enough, I've seen it twice in the field. I've seen it once in the Philippines and once in Sumatra. But both times it was in bud. There was a large bud like a cabbage. So, but I've never seen it open. So is it dioecious or is it hermaphrodite? Can someone tell me?
Ya ridanya itu. Well, we think oh yeah, dioecy. Thank you, Kunmeng. Yeah, because he also write it down again, like in the chat box. It's dioecy.
Yeah. So that would be a very interesting subject of study. So if it's dioecious, then it implies that there has been a problem with selfing, and there is some advantage to forcing outcrossing. But it does imply that there must be, when a flower is produced on a male, at the from the roots of the tetrastigma vine that it's growing on, if the plant in a tetrastigma vine is male and it produces male flowers, then there has to be a female. I believe it's fly pollinated, so there has to be a female somewhere nearby in the forest for flies to transfer the pollen. So, and I've always thought of it as being the flowers being very solitary, that you're lucky to find one. The chances of finding two from different vines at the same time seems to me to be quite small. But for the pollination system to work, does it always set fruit? If you see Rafflesia in the forest, will those flowers always produce a fruiting body, or do some of them fail to set seed? Perhaps Kunmeng Wong would comment on that.
And Kunmeng, you're welcome to open the mic if you would like to chat with Quentin.
Oh, right. So it could be monoecious. That's a fascinating thought. Of course, because it's difficult to know whether where the plant body is. How can we know whether it's just a particular individual within a vine is just producing males, or it could produce a male one year and then the next year it could produce a female? That really needs to be worked out. Yeah.
Okay, we have a lot of questions here. And the next one also from the chat box from Rohmah Supriyati in Bengkulu. Well, Bengkulu is in Sumatra. And she's asking is there any scientific effort or methods to overcome protandry and protogyny phenomenon? Thank you.
So yes, this is a very different topic, because these are both strategies of hermaphrodite flowers, but it's of course it's very like it's a hermaphrodite answer to sequential monoecy, and the one has a female phase and then a male phase. And an extreme example of this is heterodichogamy, where a particular plant only produces protogynous flowers, and another plant will only produce protandrous flowers. So they do this at different times of day. So they can't pollinate, they can't self-pollinate, but they can match each other. An avocado pear does this, and it's very difficult to break the system to give self-pollination. And I think this is this question is about whether it's possible to break a strong protogyny protandry system and in order to encourage selfing, which one might want to do in a crop system, say. And I'm not sure. I think one would have to know what the physiological controls on the floral development were. Maybe hormone treatments could delay a transition or something like that. I don't know. I'm not familiar with research in this area, if indeed there is any.
Okay, I hope that's answering the question from Rohma Supriyati. Is there any other questions? Otherwise, I'm going to ask something. I put my privilege as the chair on the last second. Thank you, Quentin. And could you please show to us again one of your slides that shows the transfer? Well, it's not the transfer, but the mechanism that has lines between the ARR 17.
Yeah.
So I think
just
yeah, just go back a couple more slides. Yes, this one. Hang on. Not this one, but like you have passed it like 3 more slides after this. One more.
this one?
No, after this one, I believe. Oh yeah, this one. Perfect.
Right. Yeah.
So this is the XY system, and based on this XY system, it appears to me that, well, please correct me if I somehow get it wrong, but for me, reading this figure, it shows that the default setting of the chromosome is can I say that the default setting of the chromosome is X, and somehow the duplication of the ARR 17 makes it into male?
Yes, it's a little bit complex. I see where you're coming from.
Yeah.
I think at one level you could say, well, the duplication of ARR 17 is required for there to be a male, because female is in both systems. But if you think about it developmentally, it's the switch is actually the feminizer gene, the pink gene there. And so if you get rid of what we did was we CRISPRed out the pink arrows, and it turned into a male because it didn't have the feminizer in anymore. So in a way, the default sex is male, and you need a feminizer to change it into female. So developmentally, the default sex is male, and you need a feminizer to turn it into female. But in this particular case, it's complicated because the feminizer is always present. So you're always going to be turning males into females, unless you have the suppressor, the blue duplication, which then can suppress it. So it's like adding another layer on top. But I think you can see that the default sex is actually male. If we think about, unfortunately, I don't have the slide here, but if we think about the ZW system, where it's like this, but it's simpler. We don't have these lines, these suppressing lines. We just have we either have one single feminizer or no feminizers at all. And so the males just don't have any feminizer. And females, they have a feminizer. So I think that makes it clearer that male is the default sex, and you just need this feminizer to make it female. That's how I see it anyway.
Yeah, well, it's so complicated when you elaborate it
I agree that for a single gene system to work, then there has to be a default sex.
Mhm.
And that default sex has to evolve at the stage of monoecy. So I think single gene sex determination can only work if dioecy has come through monoecy, because you have to do so much of the heavy lifting
Yep.
in evolution at the monoecy level. And funny enough, we don't understand nearly enough about how monoecy evolves, because everybody concentrates on dioecy. And I think some of the most exciting work in the future will be actually on monoecy.
Yeah, well, it's just beyond our current comprehension at the moment, I believe. And lifting this perspective into the higher level, and I'm not sure whether this is a fit perspective or not, but when I look at the plants as well as the animals, when I compare them side by side, well, I'm not even sure whether this is an orange, this is whether apple to apple comparison. But what I have in my mind is in animal we have like most of them, not all, with sexual reproduction, either XW or either ZW or XY, they have sex on, well, they have different sex on different individuals.
Mm-hmm. Yeah.
On plants,
so they're all dioecious. Yeah, we are dioecious.
Indeed, in the perspective of plant sexual system, in animals, including human, we are dioecious.
Yeah.
But we are mobile, and it's relatively easy to make gene transfer and recombination. On the other side, because plants, majority of them are sedentary, they rely heavily on pollinators, like wind, animals, and in my previous thought, it explained why most of them are monoecious. So it's with less energy, with less, basically you just need to invest less using monoecious flowers, like you mentioned in your first slide. But
hermaphrodite flowers.
Yes, hermaphrodite. But is this perspective equally is I'm not sure whether I explain it well, because EVO, DIVO, and plant sexual system is not really my discipline. It takes a lot to like what you mentioned earlier. It takes a lot for organism to develop sexual system, one into like animal and the other like plants, are constrained by their life form and habits just to survive, and using this different strategy like feminizer and then masculinization. That's a whole lot than our previous understanding. Do you get what I mean? It's just like, yeah.
I think so. I think the comparison.
When I look at a bigger picture, sexual reproduction is the main key of evolution, but it takes a lot of like protocols just to navigate the whole path of evolution.
Yeah.
Not even sure whether this is a question or a comment.
Yeah, well, if I can comment back, I think you're raising an important point. I mean, but I would say that at the level of sexual system, plants and animals are very similar. But at the level of the ecology of reproduction, they're very different. But I'm going to, so let's take the human sexual system, which is an XY sex determination system. It's actually like poplar, it's driven by a single gene, or largely by a single gene. There are a few modifiers, but largely by a gene called SRY, which sits on the Y chromosome. And if you have a Y chromosome, you have an SRY gene, and in humans, the default sex is female. And if you don't have a SRY, you will develop as female. If there's an SRY gene, that is a masculinizer, and it will change the default, which is female in humans, to male. And so it's very, very similar to what I'm describing with default sex in poplar. But it's the other way. In poplar, it's the other way around. You have a feminizer, and the default sex is male, and you need the feminizer to make a female. In humans, it's the other way around. The default sex is female, and you need a masculinizer, and we know the gene, it's SRY, to make a male. And so at the level of basic genetics, it's very similar. But of course, the ecology of the system is totally different. And because in plants, it's gamete transfer is mediated by wind or insects or various animals. So the ecology of the system is very different. But if you go right down to the level of molecular mechanisms, it's hard to see any difference. And so that's how I would see it.
Okay, wow, that's quite a heavy lifting, Quentin. Yeah. Oh, yeah, we still have questions in the chat box from Ahmad Zainur. Can you also determine how the methylation works for ARR17? Why and when this methylation works from your study?
Yeah. So so this is a good question. So it really matters that the duplicates are the same sequence as the feminizer gene ARR17. So when the double-stranded RNA forms, it's basically RNA of the same sequence as RNA would be produced from ARR17 itself, but it's chopped up into lots of small bits. And there is this phenomenon called RNA-directed DNA methylation, where small sequences of RNA find a target that matches in sequence, and that triggers a process of methylation, and it's called RNA-directed DNA methylation. So it's a very intriguing phenomenon. It seems to control quite a few genes in the genome that are. It's a way of turning on and switching off genes by using small RNA interference, causing the methylation of a target gene, which matches in sequence. And so I think you can see how this could come about in the genome. You just have a duplication and another duplication which is inverted, so that when they're transcribed, they form double-strand RNA. And suddenly you get the original gene being silenced, which might be lethal. But on the other hand, it might actually be useful. If it's important for sex determination, that could be co-opted to produce sex determination. So I think this phenomenon of RNA-directed DNA methylation is the key. It's a very interesting phenomenon. It's not my area. It's a very technical subject that's still being worked out in detail. But it's clear we have evidence that this is what's going on, because we can actually see the with small RNA sequencing, we've actually sequenced the small RNAs and mapped them to the inverted repeat. And then we've shown that the methylation occurs. So the inference is pretty clear that the small RNAs are causing the methylation. But we don't actually, you know, how would you really prove it? It's still an inference because we know there are small RNAs. We know that small RNAs can produce methylation. I think it's pretty solid, and that's where we are at the moment.
All right. And I think in the chat box we have one last question because it's been around like we have been doing this Q&A for like almost an hour. So I think this is the
It's been very good Q&A. Thank you all for your questions. I really appreciate it.
We are like how to say it properly, like sexually attracted to the presentation. Yeah. So from Kunmeng Wong, in dioecious plants, males and females develop not only two sexual floral morphs, but sometimes, often, the inflorescence forms in each are also rather different. That would involve just a single, few, or a whole suite of genes.
Yeah. Yeah. So I think this is what we still don't know. We can find a switch that will determine whether an inflorescence, say, will develop into male flowers or female flowers. But what all the genes that are being switched on underneath that is still largely undetermined. So we know that there must be genes to make flowers unisexual. It may involve Madsbox genes, which are identity genes for the floral sexual organs. It may not. It may involve other genes. I think it varies. But you're right. To make the ecology work, male inflorescences obviously often have to be different from female inflorescences to make the ecology work, because the male inflorescences can be throwaway structures because all they have to do is produce pollen. Female inflorescences have to be stout and strong because they're going to bear fruit. So there is this ecological reason for having different inflorescence forms. And as for the genes responsible, I think it's probably a number of genes. I think you're right, but we don't know, or at least I don't know.
Okay, finally,
very good question.
Thank you, Quentin, for all of the discussion, and thank you to all the participants who asked actively to this session. I think we have reached the end of this session, and again, because I get the privilege to close this one, I'd like to say something in bahasa to the audience. Ya, jadi terima kasih Bapak Ibu semuanya yang sudah datang, sudah bertanya dan berpartisipasi aktif dalam SOS PRB ini. Jadi nanti kami sangat menghargai pertanyaan dan juga kehadirannya, dan kita akan selalu tampilkan informasi-informasi berikutnya untuk SOS berikutnya. Again, Quentin, thank you very much. I just give appreciation to all the audience, either from BRIN and outside, either from Indonesia or beyond, who have make their time to attend this session. Again, thank you very much. This is definitely a wide-opening session and expanding our horizon about plant sexual system, and which is not so simple, and it's definitely far from simple. And the research is still ongoing, so we wish the best of luck for you and the team, especially our colleague and friend Seni. Thank you. I believe she's in a good hand under your supervision. Unfortunately, I don't see her around, but anyway, she's probably busy doing some lab works.
Unfortunately, she was due to be visiting the mountains in British Columbia today, and the cell signal said she would join if the cell signal was good enough. but the cell signal in the mountains is very poor. But I think she's enjoying herself in the mountains, which is good.
Okay.
And I would just like to say, if I can just say a thank you to you and the other organizers for the honor of being able to speak to you today, and also a big thank you for people who attended, and for the excellent questions. I learned a great deal, which is always the benefit of doing these things. I learned about Rafflesia, I learned about oxalidaceae, so wonderful. Thank you.
Yes, and we hope to see you around in person, perhaps, I don't know, in the near future. So please
That
come
would be
and visit us again. And again, thank you, for Marlena, and for the rest of the team, the SOS team that could make this session success. Okay, it's already late at night in Vancouver, and yeah, that's a lot of questions and discussion. Okay, thank you, Quentin.
Okay, thank
Next
you very
time.
much. Goodbye, everyone.
Thank you, Quentin.
Okay.